Hello, students. Okay. You can hear me, right? ECM, right? Yeah. So yeah, I'll discuss it in quality gain parakvaran. So ECM, extracellular matrix, short note. If you haven't written, we have 5 minutes. Right? If you haven't written, online people. If you haven't written, we have 5 minutes. 8. I'll give you 8 till 8:40. Okay. So, so this was a homework, right? The short note on extracellular matrix. So if you haven't written, you have about 4 minutes more to write it. So this is what I think. So starting the day by doing something active is more productive than නිකන් භාවනා කරනකොට සමහරට, right? So this is your meditation, your whole 2 years should be focused on A level. So better warm up with something theoretical. Question එකක් පටන් ගමු. So small short note. I mean, it's a part of a short note, right? So you have in a short note question, you probably have A, B, and C, 3 parts. This is just one part, short note. So target, I can't exactly remember how many points. I think about 15 points maximum. No? Yeah, about 17 points. Target about to write about 17 points. The trip. ගිහිල්ලා කට්ටිය long weekend එකේ.
Recording in progress.
Okay, let's discuss. Right, very simple short note, but like it's up to the standard of coming in the A level exam itself. Right, so at this point we are going to discuss. I mean, topics that are kind of unlikely to come for the sake of sort of studying them. But I'm going to say if something has A level exam potential, like right. So this is that kind of a topic, right? Likely to be questioned. Um, so these are the points that I sort of thought are likely to come. I mean, bring you marks, right? And before that, let's quickly revise what the extracellular matrix is. Right? What do you have? You have to have a diagram coming into your mind. Um, when I say the extracellular matrix. You can draw a diagram of your own, but you won't be asked to draw this. Okay. So, as I was saying, you won't be asked to draw this, but sometimes it helps for you to have a visual idea, right? So, what kind of cells usually have a extracellular matrix? Because they need one. They don't just have this sort of a complex structure because they need one, right? Because they functionally have a requirement for this sort of structure. What is the basic requirement or the basic function of the ECM? To provide sort of a support to the cell. So what kind of cells inherently don't have a support? Usually animal cells. Because why? Plant cells have a cell wall, which is a very stiff and sort of sturdy structure, right? Okay. So that's why animal cells have evolved to have this sort of ECM. So if you take our skin, so outermost layer we have like a plate like layer of cells. So if you zoom in, they are like actually like a snake's scales, our outermost keratin layer of the skin. It's like very plate like, right? But at a very small microscopic level. Right? Underneath that, so that is called what? Our outermost skin layer is called what? Epidermis. Epidermis. Epidermis එකට යටින් ඉන්නවා dermis කියලා එකක්. Right? Then dermis is connected to this sort of a extracellular matrix. Then there's another cell layer, or we say a tissue called the connective tissue. Right? Connective tissue කියලා. We call it the areolar tissue. So if you like remove our skin, you know our skin can be removed like a suit. Right? අපි flay කරනවා කියලා හැම ගහනවා කියලා, right? Have you seen in butcher shops? Like they can easily remove the skin, right? There are in motor traffic accidents, if a person, so we are doing forensic these days, right? Adhikarana, right? So we have to analyze these accidents, right? Motor traffic accidents. So there are accidents that are called degloving accidents. What do you call degloving? Degloving injuries. What is degloving? Glove එක ගලවන. So if a person sort of drags a bike එකකින් වැටලා පාර දිගේ ඇදිලා ගියොත්, so relative to their leg, their skin gets disconnected. Do you understand? මේ skin එක underneath tissue එකකින් disconnect. So the entire skin is sort of separate from the muscle now. It's like a suit, like a glove එකක් ගලවලා වගේ degloving. I won't show you any images because you will stop the class for showing graphic things. Degloving. So that is kind of reduced by having this sort of a connective structure outside cells, particularly in animal cells. Okay, that is the basic function of extracellular matrix. So what are the things that you need to know? What is the main composition? This yellow, no? Yellow color component. That is what? Collagen fibers. So so there was a question this week as well. Collagen is what? We learned under proteins. It's a collagen is a protein and having what sort of organization of protein? Primary, secondary, tertiary, or collagen is quaternary. Because why? It has 3 protein subunits, right? It's like a rope on the kambayak wage. 3 protein subunits wind around each other to form collagen. That is the main composition. Because why? Collagen is very strong. Okay, but here, because it is embedded in this sort of mesh or network that we call as proteoglycan, we consider it as a glycoprotein because it's very closely associated with what? Carbohydrates, right? So here, that's why in the resource they say most abundant glycoprotein is collagen. Okay, then you can see these sort of branching structures. Those are called proteoglycan complexes. Proteoglycan complexes have a central, can you see, it's like a branch. The central sort of the branch is a polysaccharide, and from that polysaccharide there are small protein, let's say small branches, and there you have like leaves lage carbohydrate, small pieces of carbohydrates, chains of carbohydrates. That is the basic structure of a proteoglycan complex. You don't need to know that. So you have to say collagen is embedded in a proteoglycan network that is secreted by this guy. This is the big cell, no? Getting in a big cell. And then these collagen fibers, you can see small green colored proteins that are called fibronectins, right? Can you see? If I zoom in, you can see, right? Fibronectins. So the collagen or the proteoglycans are connected to the plasma membrane by small protein pieces called fibronectin. This is what? It's actually, I mean, it's an integral protein because it is anyway embedded, but as it is going the entire thickness, so it is also what? It's also a transmembrane protein. Integral protein, it's a transmembrane protein. And to the inside, so the cell inside, this is the interior of the cell. In the inside of the cell, there is another supportive structure. What is that? The skeleton of the cell, cytoskeleton. So you can see the cytoskeletal elements are also connected to this integral protein. Integral protein is connected to a fibronectin. Fibronectin is connected to the extracellular matrix. That is the sort of whole interior connection. Sorry, inside and outside connection, right? Those are the ideas that you need to have. Clear? So let's move on to the marking scheme, right? So mark your essays, sorry, short notes. No need, right? Those introductions, you don't carry marks. They have an elaborate extracellular matrix, right? Main components of the ECM are glycoproteins and other carbohydrates. Mark 2 marks. Molecules secreted by the cells. Okay? Mark your essays. Sorry, short note. Glycoproteins and other carbohydrates. Carbohydrate containing molecules secreted by cells. Then the most abundant glycoprotein, 1 mark, is collagen, another mark, which forms strong fibers outside the cell. Following people, you can hear me, right? Yeah. The collagen fibers are embedded in a network, embedded point, network woven out of proteoglycan point secreted by the cells. Collagen fibers are connected to integral proteins, 1 mark, of the plasma membrane, 1 mark, right? You have to say via fibronectin point, right? Right. Then moving on to the functions, its main function is a protection role, so forms a protective layer, links ECM and the cytoskeleton, influences cell behavior by involving chemical and mechanical signaling. Because of the physical connection between the cytoskeleton and ECM, there is direct physical connection, no? And also I have told you, your microtubules in the cytoskeleton is like a railway track, no? Inside the cytoskeleton, the microtubules act as a railway track. These motor proteins can take different vesicles here and there in the cell, right? So through that railway track, the motor protein brings something close to the plasma membrane, and through a transmembrane protein, that something vesicle goes out of the cell, and through the extracellular matrix, it can be sort of transported out. So through the cytoskeleton and through the plasma membrane and into the extracellular matrix, there is also chemical connection, right? Through the, let's say, exchange of these sort of vesicles, right? So that's what they say, chemical and mechanical signaling. Okay. Okay. I need to know out of 17 how much you got. Online people, send me your marks. Okay. Right. So today my plan is to try to finish cell division. Mitosis I have started with the introduction, right? Interface I talked a little bit, so I'll just quickly go through that. Anyone who's brand new, you didn't get the main tube, you got right? So there's also anyone else? You have the main tube, right? Yeah. So this is just a this that one only covers the cell cycle. Okay. Right. So today, if I am not able to cover cell cycle today, we'll take a small time from next week and quickly cover it. And next week, I plan to start the next important section, metabolism. So this is the new tude for that. That is going to include what? Photosynthesis and cellular respiration. Very question heavy areas. Essays, structured essays, pass paper karamban, right? So in the tude, actually, there's about categorized past paper questions for about 4 pages, right? Within those 5 years, there has been so much questions from that photosynthesis and cellular respiration part alone, right? So we are going to sort of pay a lot of attention and do a lot of questions, right? So this tude I'll give next week. That is going to cover photosynthesis and cellular respiration. So I'll probably take about 4 weeks, but I try to, let's see, right? I'll try to plan to, I mean, within this month, because next month I have, together with the entire institute, there's going to be sort of a, let's say, mock exam to prepare you for the first term test, right? Because at the end of the day, that's what's going to give you confidence, right? School term test වල marks තියෙනවා. ඉස්කෝලෙන් ඉගෙන ගත්තේ නැතත් ඉස්කෝලේ ඉගෙනුවේ නැතත් ඉස්කෝලෙන් term test එකක් තියෙනවානේ. right? ඒකට යන්න ඒකට ගිහිල්ලා ලකුණු ගන්න අරන් ටීචර්ට පෙන්නන්න right? එහෙම ඉෂුව එකක් දානවා නම් පෙන්වන්න මාක්ස් දාලා එතකොට කියන්න බෑ right? අපිටත් උනේ අපි බැන්න ඉස්කෝලේ ඉන්න කාලකන්නිම සෙට් එක ඉස්කෝලේ ඉන්නේ ඇවිල්ලා ලැබ් එකට පැනලා මොනාරි විකාරයක් කළා යනවා එහෙම කියනවා ඊට පස්සේ රිසල්ට් කියන්න ගියාම කියන්නේ මොකද අපි දැනන් හිටියා ඉස්සර ඉඳලම ඔයගොල්ලන්ගේ මෙතඩ්ස් වලින් වැඩේ කරගෙන ගියා right? That's the story. So, I mean, I'm not saying to have sort of a rivalry with the school, but there are teachers that's going to support your type of learning, right? Class filling they are going to supplement you. We had a physics that helped us with our class tudes because he saw this is a good tude. Mechanics textbook mechanics questions study textbook कहती objective physics right එම big book yeah ඕනෝකෙන් අපිට questions කරා yeah ඒතර යා theory උගන්වන්න ගියේ නෑ අපි අහන් ඉන්නේ නැහැ කියලා දන්න so get the maximum support you need from school do the practicals physics practicals here school lab එකට පැනලා ඒව කරන්න because at a maths class you're not going to get that opportunity no traveling microscope එකක් දකින්න chance එකක් හම්බ no right so that's up to you to be strategic තරහ කරගන්න නැතුව get the maximum benefit so anyway that's the sort of immediate plans that we have we are going to finish second lesson I'll try to finish within this month and then we are going to start the third lesson evolution for that I'm going to take a bit of time because I'm going to be doing part of the fourth lesson in the third lesson as well okay so that is sort of the structure I have got used to anyway right so today let's finish cell division let's try to finish cell division I think last week we took the introduction to mitosis right mitosis quick revision what are the two phases of mitosis right what is it what are the two phases yeah if we draw it's a cells undergo a cycle right what is a cycle there is no beginning or there is no end right? We can say the cell cycle begins here and ends here because it's a continuous process. So we mainly divide it into what? Two, let's say, if it's a pie chart, the biggest part is what? It's called the interface, no? So usually from up to here to about here, right? About 90% of the cycle is interface. Interface, right? About 90%. A very small piece is called the M phase or the mitotic phase. Okay. What are the three parts, let's say, subdivisions of the interface? Gap 1, G1, synthetic S phase, gap 2, or second gap phase, right? And then, I mean, so M phase we are subdividing into other sections, prophase, metaphase, anaphase, or more, subdivisions theorem. So we talk basically about these, right? How the cell prepares to this M phase. So M phase it starts another new interface, and when the interface is over, it starts a new M phase, like that. Whenever the cell has divided, cell at home in the lab, there was a cell, and it gets slightly bigger, then it divides into two cells, sort of similar to the original size. This is basically what happens in mitosis. No? So the cell is actually spending a lot of time in this growth phase, right? So almost all the growth happens in the interface, right? Normally non-dividing cell it's in this sort of interface. It's growing, becoming bigger as a normal cell does. Then suddenly when it is kind of had enough growth, it divides in the sort of symmetrically into two equal cells, which these are called what? This is called, the original cell is called the mother, no? Usually the mother cell. The mother actually gives rise to another, no? These are both called daughter. But if you sort of look at it closely, one of them is still the mother, no? From the mother, there was another one, right? So these are both daughters. Really? We say the mother cell divided into two to give rise to two daughter cells, right? Clear? Okay. So quick revision of what happens in the interface. What are the things happening in G1? Take out your resource books, okay? Online people, you can't hear me. Online people, not clear. How about now? Not clear. About now? Still no? How about now? Yes. Now okay. Right. Okay. Sorry about that. Right. Okay. Page number 39 we start. So interface is the longer phase. It's about 90% of the cell cycle. Highlight those points. Interface has three phases, G1, S and G2. These things we discussed last week. So I'll quickly go through. Okay. Synthesis of proteins. What happens in G1? Synthesis of proteins and production of cellular organelles. Right? I told you, this is like a normal cell. Yeah, in the future I'll divide. But he's anyway growing, right? So he's doing like you, from a baby to adult, the cell is growing, right? So that's what happens in the first growth phase, right? First gap phase. Proteins essential for the S phase are produced. So G1 to they are going to move to S. So they are preparing for the S phase as well. S phase is very important. There's a phenomenon called DNA replication. DNA photocopying. Right? තියෙන DNA ටික will be copied into exact another set of DNA. Because why? එක සෙල් එකක DNA නොදී බෑනේ. නිකන් අර දෑවැද්ද නොදී ඩෝට වේවුවා වගේ, right? So this cell has to prepare another set of DNA to give to the daughter. That is what happening in the S phase. DNA photocopying, right? So highlight that DNA replication occurs. Synthesis of histone proteins take place. What is the importance of this histone? There's a thing called DNA packaging, right? It's a bit difficult to understand. So how does DNA look like in a normal non-dividing cell? Cell එකක් divide වෙන්න හිතලවත් නැති වෙලාවක. How does DNA look like? If you take a closer look at the nucleus, it's like a මෙහෙම ball of string, right? නූල් බෝලයක් වගේ තියෙන. DNA, right? It's messy. Why is it messy? Imagine this DNA as when you are studying, right? A level වලට close තියෙද්දි ඔයගොල්ලන්ගේ මේසේ නිකන් book rack එකේ ලස්සනට resource book ටික, review books ටික එහෙම ලස්සනට pack කරලා තියන්නේ. They're everywhere, no? Because why? You need to quickly access them, right? So in a normal working cell, DNA ටික ලස්සනට pack කරලා තියන්න බෑ. It is everywhere, right? That's why the chromatin is like a messy ball of string, because it is constantly being used. And when you are reading something, you are taking information, the DNA is controlling different functions, right? So this is chromatin in a normal non-dividing cell. What is the magnified view of a chromatin? This is, I told you, it's like a string, but the chromatin, so this is, if this is the DNA, it comes like this, and there's a friend that helps this chromatin to package, that is called histone. Right? 8 histone proteins, they are like ball-like proteins. So this DNA wraps around this histone twice, right? දෙපාරක් මෙහෙම wrap වෙනවා. And then it goes like this, and there's another histone, 8 histones like this, right? Right? And it wraps around twice again. Right? This is how chromatin looks like in a magnified image. This is chromatin. But when you are trying to give this daughter cell DNA, so මෙහෙම chromatin තියෙනවා. This chromatin has to be, now it is been doubled. So you have double set of chromatin, but ඔහොම නිකන් මෙසීම් නූල් බෝලේ වගේ දෙන්න බෑ. Right? It has to be nicely packaged. No? Imagine you are done with A levels and you are giving all your notes to some like a sister or a brother. නිකන් ඔන්න මේ ඇවිල්ලා ගන්න කියලා දෙන්නේ නැහැ. You are going to nicely pack them into a box or something, right? That is what the cell is going to do. It is going to nicely package this chromatid into a very stable structure. Right? නිකන් කිසිම DNA ටිකක් missing වෙන්නේ නැතුව දෙන්න පුළුවන් stable structure එකක් හදනවා. That is called the chromosome. Right? Chromosome. Right? So in your resource book, they just assume that you have this knowledge, right? What is a chromosome? What is chromatin? They just assume, right? So this is a very let's say compact structure. So මම මේ දෙකම එකම scale එකෙන් යනදට this entire chromatin is going to be packed to a very small structure. Okay, that is because it's very tightly packed, right? පොට්ටික ලස්සනට box එකකට pack කරලා ගිහලා දෙනවා. Right? So this is chromosome. The biggest thing that happens in one of these M phase, the beginning of this M phase, is chromatin becomes chromosomes. Okay, that is going to help this process of giving the two daughter cells the DNA nicely. Okay, that idea you need to have. Right, then S phase, so that's what happens. DNA replication occurs and synthesis of these histone balls also happen. Why? Because you need to pack them, no? Right? Histones are to pack already. G2, again, it's very similar to the G1, right? Protein synthesis, cellular organelles. Protein is essential for the mitotic phase also be synthesized, right? So the cell is now preparing for the next step, that is mitosis. Duplication of centrosomes. I talked about this last week. What are centrosomes? Two. Yes, online people. What are centrosomes? Two perpendicular bundles of centrioles, right? Okay. Two perpendicular bundles of centrioles that are usually found in these two edges, right? Because the cell is going to divide into two, only one set of centrosome is not enough. You need two, right? You need two centrosomes. That's why they are going to be doubled. Okay. Then there are cell cycle controlling checkpoints available at G1, G2 and M phases to ensure that the cell is ready for moving into upcoming phases of the cell division. Right. So where do you have these checkpoints? G1, there's a checkpoint, right? They are going to check the cell and sort of here before M phase you have a checkpoint and sort of beginning of M you have check. Okay. I told you the importance of this, right? They are going to ensure that the cell is ready to divide. Simply put, okay. They are going to ensure if the cell is ready to divide. What is going to happen if a weird cell divides? Like a weird one. Yeah, හරියට ලෑස්ති වෙලා නැහැ divided. His DNA is not properly copied, right? He has not gotten bigger enough. Sometimes his DNA is damaged DNA. If an abnormal cell like that divides, that could be the beginning of what? An abnormal cell division. What is an abnormal cell division? Yes, it's what we call as a neoplasia, like a new cell growth that can go two ways, right? It can create a ball of cells that is going to stay at one place. That is called a tumor. Or it can be very invasive. Invasive, නිකන් අර Russian army එක වගේ. They can be invasive and try to go to other places of the body. That is called what? A malignant cancer. So that can go two ways. Whatever the outcome, that is going to start with one weird cell, right? So that's why not just one, not two, but three checkpoints are there. Okay, very important for survival. Okay. Yes, cancer. Okay. So please remember G1, G2 and M phases. Yeah. If not, does not receive a go ahead, right? Then check now. Then bila dekala api checkpoints ala check and drivers la. What are we checking? If the driver is suitable to drive. So here, what are we checking? The cell is suitable to divide. Similar thing, right? එතකොට බීලා කියලා අහුනොත් සෙල් එක abnormal කියලා අහුනොත් මොකද කරන්නේ? නවත්තනවා. ලයිසන් එක ගන්නවා පොලිසියට එක්ක. Right? It's like that. If they find out if the cell is not ready, they are going to force the cell into a non-dividing stage. It could happen at either checkpoint, right? G naught කියලා ඊළ හිරේ දානවා වගේ. Right? Very බහිනකන් remand එකේ ඉන්න හැරි මොකක් හරි that kind of thing, right? So if they do not pass at these checkpoints, they are going to go into a non-dividing stage called G naught. Most of the cells in the human body are in the G naught phase. What are the examples? Nerve and muscle cells. So I told you, nerves and muscles are what we call as stable cells. Stable, right? For example, your nerves, particularly in your brain, they are the same number that you had when you were born. වැඩි වෙන්නේ නැහැ. Right? They can become bigger. They can create more connections. Right? When a neurons connect, they can become more complex, but the number is the same. Right? Number of neurons in your brain is the same. Right? And also muscles. I told you, right? How bodybuilders become bigger. Do they increase the number of muscle cells? Very, very small number. But what they actually do is, because of the stress they put on the muscle, right, by working out, the muscle is going to increase in size. That's why. Okay. So that's why these bodybuilders in the off season, if they stop taking their protein intake and reduce the amount of exercise they do, they are going to lose the muscle size. Because why? If the muscle cell number increased, reverse current bear, right? But the size increase can go back, right? That's why. So these muscle cells and nerve cells are stable. So they divide once. Ammage bade indeddi your nerve cells and muscle cells are created, right? You were initially this ball of cells, aching they were created, then they grew, right? In the G1 phase, okay, they grew into sort of a normal size, then they exited. Mamanang I divide when. That's what happened to these stable cells. Okay, clear. That's what you mean by this G naught. Okay, moving on. So that is interface done. Any questions? Last week we wrote a short note from G interface. No? So I think the understanding is fine. All right. Any questions? Yes. No, right? Then we can move on to where the actual sort of we have the date. Actual sort of events of interest happen. හැබැයි ඒක cell cycle එකේ percentage එකක් ගත්තොත් it's actually about 10% of this whole timeline. No, very small duration, but many significant things happen. Okay, right. So mitotic phase or the M phase covers about 10% of the cell cycle. Come to the resource. Let's highlight and let's discuss. Okay. Definition of mitosis. Highlight. Online people, I want you to have the resource book ready. And if you haven't got delivery of the tute, have the PDF. I have sent it to the group, right, of the main tute. Please have those two at the ready. Online people. Okay. What is the definition of mitosis? It's the nuclear division which gives rise to two genetically identical daughter nuclei from a mother nucleus. Highlight the definition and then underline daughter nuclei from a mother nucleus. Right? Very, very important phrases. Why are we saying it is just a nuclear division? Because actually, මෙතන පොඩි subdivision එකක් තියෙනවා like this. There's a very small subdivision here that is called what? Cytokinesis. That is not actually part of mitosis. Cytokinesis. Right? So when this mitosis is associated with cytokinesis, you have what? Two daughter cells. But if only mitosis happen, you have only two daughter nuclei. Nucleus දෙකක් විතරයි හැදෙන්නේ. But there is something, if something called cytokinesis happens, you're going to have what? Two cells. Okay. There are 5 stages: prophase, prometaphase, metaphase, anaphase, telophase in the order. Yeah. Okay. So what are the phases in this M phase? You have inside this M phase, you have prometa, ana, telo. Okay. 4 subdivisions in the M phase. All right. Any questions? No. Preface. Shall we discuss? Okay. Page number 41. Preface. Right. So whenever we are discussing a phase, I'll tell you to put numbers. A phase එක ඇතුලේ there are few things happening. Right. So I want you to remember each of the things happening as 1, 2, 3. Okay. So ඔයගලන්ට මතක් වෙනවා when you are studying, 4 things are happening in preface. Right. නිකන් paragraph එකක් මතක තියාගන්න බෑ. So I'm going to tell you to put numbers, then you highlight and put a number. Okay. Right. Chromatin fibers get condensed by shortening and thickening and transform into chromosomes. This is called what? This process is called what? It's called condensation. 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 Chromatin, messy ball of strings become a nicely packaged, like a package, for ready for delivery, right? So that's number one. Put number one there. What is the first thing that is happening? Chromatin fibers are condensed by shortening and thickening into chromosomes. Then keep reading. As a result, chromosomes will be visible through the light microscope. Right? So, if you remember when we studied nucleus, what did we say our DNA looks like? Our chromatin. What did it say? Diffuse mass diffused. Abhi individually strings pain nahi. Not even from the light electron microscope. Because why? It's a ball of string, right? But when they condense into chromosomes, they are very clearly visible because they are very compact structures. Okay, so they are visible under light microscope also, right? Light microscope also. In your practical guide, there's a practical uh, that is called observation of cells in different phases, right? I think I have some diagrams. I'll show you. Okay. I think in your school labs you have this laminated diagrams එහෙම තියෙනවා. ඕවා නිකන් උස්සලා ගිල්ල බලන්න. What are the different phases, right? So, right, moving on. Nucleoli get disappeared and chromosome appear with two sister chromatids attached at the centromere. Right. Lot of things to explain there. Right. So I'll erase this. We are done with interface. What are the things in the interface you need to know? Three parts: G1, S, G2. Growth happens in these two. DNA replication happens in S. There are checkpoints at G1, G2, M. If they don't pass the checkpoint or receive a go ahead, they enter into a genome. Finish. Okay. So, what are these sister chromatids? Sisters. So, I told you in the S phase, this DNA is actually going to double. Another ball of string will be produced, right? Exact copy, right? Because that's what happens in DNA photocopying, DNA replication. That is going to also undergo this condensation, and that is going to also produce what? A chromosome. But the thing is, these two chromosomes are not going to stay separated. They are going to be attached at what we called as a special midpoint. No, it's not exactly a midpoint actually. So it's called the centrosome. Centrosome. That centrosome actually is formed due to a part of DNA in this chromatin. එතකොට මේ DNA එකේ there's a part that will become the centrosome. Right? So that blue color part is actually present here. Centrosome sequence කියලා කියනවා. Right? So the main function of the centrosome is actually to attach the two sister chromatids. So the two sister chromatids are attached at their centrosomes. So when these two chromosomes are attached at their centrosomes, they are called what? Sisters. Sister, not chromosomes, chromatids. Okay, called sister chromatids. What are the features of these two? Exactly the same. They are exactly the same, right? Same copies of each other. Okay. Then there are names. අපි මේ arm එකට කියනවා, so there's this is called the upper arm, this is called the P arm, this is called the Q arm. Right. ඔය වගේ එක එක තියෙනවා, right? So centrosome, at the centrosome they are attached. Clear? So centromere, no? Sorry. This is called centromere. Right? Centromere. Yes. Centromere. Right? They attach at the centromere. So it's a mistake. I still do, right? Right. This is called the centromere sequence and they attach at the centromere. Right? Okay. So put number 2, nucleoli get disappeared. That is the second thing that is happening. Nucleoli get disappeared. Thank you. Okay. Nucleoli get disappeared. That is the second thing that is happening. What is nucleolus? You can remember, right? If you take the nucleus, which is a double membrane bound organelle, right? It has this nuclear pores and there's a condensed part that is other than the chromatin, that is called the nucleolus, right? Nucleolus. What are the functions of the nucleolus? What are the functions of the nucleolus? There's a main big one. Nucleolus, not the nucleus. Yes, online people. Making of what? Nucleolus makes what? Ribosomal RNA. No? rRNA is made by nucleolus. rRNA. So, resource book put number 2, nucleoli get disappeared. There is a second thing that is happening. Nucleoli get disappeared. Then chromosomes appear with two sister chromatids attached at the centromere. Centromere. Okay. The two sister chromatids. The two copies. These are chromosomes. But when they are connected at the centromere, they are called what? Sister chromatids. Then chromosomal arms of the sister chromatids are attached by special proteins called cohesin, right? So at the connection point, you have a protein that is like a temporary glue. It is water soluble, right? So this cohesin is a reversible glue that will later become, it will later go away, right? So for now, temporarily, the sister chromatids are connected by this cohesin protein. Highlight the word cohesin. Okay. Highlight the word cohesin. Formation of mitotic spindles begin. What is the spindle? අපිට spindle එකක් කියලා කියන්නේ spindly structure එකක් කියලා කියන්නේ it's like a let's say apparatus එකක් වගේ එකට නේද spindle එකක් කියන්නේ. So your mitotic spindle is a sort of an internal robotic arm, controlling system that is going to mainly involved in moving these sister chromatids. It has several parts, right? This spindle has several parts. So if you take the cell like this spindle arises mainly from the centrosome. Centrosome. The two centrosomes. Why do we have two centrosomes? Because they got duplicated. Which phase? Which phase? G? G2? Yes. Because now we have two. So from these two centrosomes, there's going to be what? Centrosomes are what? Two centrioles. Centrioles are composed of what? Bundles of microtubules. Microtubules are going to arise from these two centrosomes and sort of connect with each other across the cell. Like this. These are called spindle microtubules. Right, I'll label them. I'll draw the cell in a different color. Right, spindle microtubules. So, maybe the microtubules that are connecting the two centrosomes are called spindle microtubules. All right. So, this is what? Centrosome. And there are very short microtubules that are going sort of the opposite direction, like this. Pitipasara. They are very short, but they are anyway part of the spindle. They are called what? Asters, right? Aster microtubules. Okay. Any questions? Right. So that's number 3. No, spindle is formed by accumulated microtubule complex, which includes centrosome, spindle microtubules, and the aster. Highlight that. That is the third thing that is happening in prophase. Spindle is formed. Highlight that, online people. Spindle is okay. Then centrosomes move towards the opposite poles of the cell due to the lengthening of the microtubules in between them. So these microtubules that are long ones, or the spindle microtubules, connect with each other. Then they push the two centrosomes to the far ends. We call the poles of the cell. Cell like poles of the push current, right? So that is the end of prophase. Any questions? That is the end of prophase. So take out your tubes now. Let's write a few things. Cell cycle in the main tube, this page. Cell cycle, right? Online people, I have put the PDF in the group. Do you want me to put the PDF in the Zoom chat as well? Anyone who was not able to download it? You have the main one, right? You have the main one, right? Yeah, it's there. This is just a one for the people who are joining this month. Okay. Right? This page, cell cycle. So I have given you a small diagram to understand what is this binary fission. Okay. Binary fission is a word that we are going to encounter. Can you see on the top right corner of the tube? You can. I have put a small diagram that is describing binary fission. Binary fission is like a primitive form of mitosis, right? ඒගොල්ලෝ full mitosis unlock කරලා නැහැ, right? Usually in prokaryotic bacterial cells, this binary fission is happening. It's not very well nicely regulated. හරියට equally DNA is getting into the two cells. Those things are not properly regulated in binary fission. Okay. So, but even though it's not in your syllabus, you are going to come across these words in several places in the resource. So that's why I put that diagram. So yeah, it's a very primitive form of proper mitosis, right? Okay. So on the other side, I have put a sort of a summary of what is going to happen, right, in mitosis. How so we have taken an example of one chromosome, right? We are assuming that the cell that is dividing is only having one chromosome. So if you take a human cell that is dividing, how many chromosomes are we going to have? මේ වගේ 46. But after replication, how many are we going to have? 92. So 92 are going to go into the two cells. So daughters are then having 46, 46. Happy. Right? That's what going to happen. Clear. So go home and read that. Then mitosis topic. Okay. In the tute, please write down the definition of mitosis under the topic. What is mitosis? Write down. Yes, the nuclear division. The nuclear division which gives rise to two genetically identical to two genetically identical two genetically identical two genetically identical daughter nuclei from a mother nucleus. From a mother nucleus. Okay. Then on the right side, sorry, underneath that we have a diagram. Right? We have a diagram. What do you think that diagram is? What are the things that you can see in that diagram? Yes. So for the clarity's sake, we have removed all the other organelles. නිකන් මේ cell එක divide වෙනකොට mitochondria හැම නිකන් එළියට යන්නේ නැහැ. Right? They are still inside. Right? But for clarity, we are not thinking about other organelles. So what is the only organelle that we are thinking? Nucleus එක විතරයි. Right? Because it's what? It's a nuclear division, no? But for your interest, your mitochondria and chloroplast undergo a similar thing. Why? Because they have their own DNA. Ne? මතකද? In chloroplast and mitochondria, they have their own DNA. So when the cell is dividing in those gap phases G1, G2, mitochondria are increasing in numbers. Chloroplast are increasing in numbers. So how are they increasing? They are also dividing in a process that is similar to this binary fission. Okay, so that is something additional. So first diagram, that is what? This one. What do you think it is? Prophase, no? Prophase. For the topic, that is prophase. Then label. What are the things that you can see in this diagram? What are the things that you can see? You can see what? From outside to inside. What are the things you can see? You can see the two centrosomes, yellow colored. Can you see the two centrosomes? So label. Online people, you are following me, right? This diagram. Yellow colored, you can see the two centrosomes. Why? They got duplicated, no? Two centrosomes are there. Label. And can you see the sister chromatids inside the nucleus? Purple color nucleus. So what is that purple color sort of shell? Nuclear envelope, no? So label, put an arrow to the purple color shell. That is nuclear envelope. Label that. And inside, can you see a small another purple color ball? Small purple color ball like structure. What is that? Nucleolus. Then what else can you see? You can see the sister chromatids. Put an arrow inside. Can you see? They are connected at the centromere by cohesin protein sister chromatids. Put an arrow there as well. Sister chromatids. Okay. Then write down there. There's space I have given. Right. There's a space I have given. Four things that are happening in the prophase. Very briefly. Right. First one. Put number one and quickly write. Chromatin get condensed to chromosomes. Number one. Chromatin get condensed to chromosomes. Chromatin get condensed to chromosomes. What is the second thing? Put number two and write. Nucleoli get disappeared. Nucleoli get disappeared. Okay. Third one. Third one. Mitotic spindle is formed. Mitotic spindle is formed. So, alright, write this under the first diagram only. Okay. Second diagram we have to write some other things. Okay. That is for metaphase. Okay. It's fine. Okay. So, third thing, mitotic spindle is formed. Okay. Fourth one, centrosomes move to the opposite poles of the cell. Centrosomes move to the opposite poles of the cell. Okay. So in your Campbell biology, there are a few diagrams, but the online version doesn't have. It's not very. I mean, it's not very clear. So that's why I took these diagrams from the software, right? Okay. Clear about prophase? Very important phase, right? It's all about the preparation. Okay. Moving on to pro-metaphase. Pro-metaphase actually even in the medical faculty also in anatomy, one of the first things we learn is the cell cycle. We don't learn about what a phase called pro-metaphase, right? That is something only found in the Campbell. Okay. It's a in-between phase between what is the previous one? Prophase. What is the next one? Metaphase. Pro-meta. Right. So let's read about. Repeat the fourth one. Centrosomes move to the opposite poles of the cell. Yes, thank you. Yeah. Then there's before that there's a small point there before prometaphase. Centrosomes or centrioles are not available in plant cells. Highlight that MCQ point. You know this, right? When we talked about the centriole, when we did cell structures, okay, centrioles. එතන තියෙන point එකක් තමයි centrioles are not found in plant cells. So if there are no centrioles, there won't be centrosomes, right? But anyway, in plants, there is what we call as an accumulated microtubule complex. It's like a bundle of microtubules that is not properly organized into this sort of centrosomes, right? It's a bit messy. So have you seen this in this Perahara? There's a dance called Vevel Natumukilati. There's a central person who's holding into a bundle of this bigger Vevel, and there are people running around. Right? Have you seen that? So the centrosome is like that. Right? He's the central person who's holding all these what microtubules අදට අල්ලන් ඉන්නවා. So they can move here and there, right? That's what is the centrosome is doing, right? It's like properly nicely holding in them in an organized way, right? But in plant cells, there's no such thing as centrosomes. So it's sort of a messy structure, but anyway, a spindle is formed in plant cells, right? A spindle is formed, but it's not very organized like in animal cells, right? So under there, if you can draw this very rough diagram, this is what the spindle looks like. There are two centrosomes. There are spindle microtubules that are connecting the two centrosomes. Then there are these short microtubules called aster. Quickly draw this. Quickly draw this. Now the spindle, okay. Very rough diagram. What I want you to know is what are the three components of the spindle? You have two centrosomes, spindle microtubules, and the aster. Right? In the structured essay, they can ask you what are the components of this spindle. So this is the mitotic spindle. To be more specific, this is mitotic spindle. Right. Okay. Right. Metaphase. First thing that is going to happen, put number 1, come to the resource book please, page number 41. Prometaphase. Very first thing that is happening, nuclear envelope fragments. Put number 1, highlight nuclear envelope fragments. Okay. Chromosomes get even more condensed. Number 2, chromosomes get even more condensed. Put number 2, chromosomes get even more condensed. A special protein called kinetochore attaches the sister chromatids of each chromosome at their centromere. If you like, you can draw this, but I forgot to tell you. Last page. Similar. Chromatin gets doubled. They become condensed into what? Into chromosomes. Chromosomes get attached at the. Sorry about that. I couldn't ask you to draw, right? It will be helpful that if you can remember this, no? I'll give you 1 minute, quickly draw. Nucleus from chromatin to the sister chromatids. Yes. What are the things you need to know? They are attached at the centromere by cohesin. මේ PQ worms කියන ඒවා ඔයගලන්ට ඕනේ. This understanding of the chromosome is going to be very important in future lessons such as molecular biology, genetics, වගේ ගොඩාක් එනවා. Particularly molecular. If you like, you can also draw this. This is what a chromatin looks like magnified. මේකට අපි කියනවා beads on a string here. Beads on a string. Beads කියන්නේ මොනවද? පබළු නෝ. Quickly. Okay. Questions? There's an accusation. I raised the board very fast. So these days I'm in the pediatric ward, and from babies you get diseases very very quickly, right? Because when you want to do anything to them, you have to touch them a lot. බාලට මුකුත් කරන්න බෑනේ වඩා ගන්න තුර. So mask දාගෙන ඉන්න බෑ බය වෙනවා. So I'm having a sore throat. So pediatrics is nice. The patients are very very nice, right? Because when they have a problem, අඬනවානේ. ප්‍රශ්නයක් තියෙනවා නම් pain එකක් තියෙනවා අඬනවා. So if you have given a solution, treatment එක වැඩ කරනවා නම් අඬන්නේ නැහැ. Simple, right? The ආච්චි කෙනෙක්ට වගේ if you are treating එක problem එකක් solve කරනකොට there's another one, right? පපුව රිදෙනවා කියලා ආවම පස්සේ when you treat whatever, if there is a heart condition, then ධනිස් රිදෙනවා, right? කොන්ද රිදෙනවා, arthritis එක. එහෙම there's no end to the list of problems. You take a baby, ඕනම පොඩි බබෙක්, if you make them better, they are going to smile. Very simple. But there's a very high risk in it, right? Neonates, චුටි බබාලා එහෙම very careful වෙන්න ඕනේ. And usually in Sri Lanka, the pediatricians are the Sarama doctors, right? I don't know why. Pediatric consultants are the most strict people. I don't know why, because maybe they are under stress or something. Okay, done. Moving on to prometaphase. Okay, so in the tute, the second diagram I have planned it as prometaphase. Right? So let's go through what is in the resource first. Prometaphase, first thing, nuclear envelope fragments. Number two, chromosomes get even more condensed. A special protein called kinetochore attaches the sister chromatids of each chromosome at their centromere. Right. I'll erase this. I'm going to keep the two sister chromatids diagram, but this one I'll erase. So basically, what is going to happen in mitosis is we are going to separate these two sister chromatids and give it to two cells. Dekata galwala, we are going to give it to the two daughters, right? That's what we are going to do. They had a house. The father is going to build a similar house like it, right? And then give it to the two daughters. That's what going to happen, right? To do this, who are the sort of motor apparatus involved? Who are going to actually deliver the two sister chromatids to the opposite sides? That is your spindle microtubules, right? So your spindle microtubules are going to come from either side and attach to this centromere by another special protein called what? So yes, that is called what? Another special protein called kinetochore. So still the blue color is the centromere, but I'll draw an arrow like this. Centromere is what? Is sort of where the two arms of the chromosome is connected. And the two sister chromatids are connected by cohesin. And the two sister chromatids are connected to what? Spindle microtubules by what? Kinetochore. What do you mean? Apparatus have a visual idea, right? Spindle, this is a spindle microtubule. 2. මේ පැත්තෙන් ඉඳලා එක්කෙනෙක් කරනවා. Spindle, right? This is a magnified view. If you take a whole cell, for example, a human cell, මේ විදිය sets කීයක් හැදෙනවද? How many sets like this? How many sets like this? 46. Because why? The two sister chromatids are going to be separated and මේ පැත්තට 46ක් කරනවා. මේ පැත්තට 46ක් කරනවා. You get two full cells. Okay, got it? Then I'll do another diagram that is going to, then the next story is that, so if we take, this is the dividing cell, right? If this is the dividing cell, there's going to be sister chromatids getting connected to these microtubules, like, so these are the sister chromatids, right? Sister chromatids that are getting connected to these microtubules, right? But the thing is, there are microtubules that are not actually connected to the sister chromatids. So this is a microtubule. So these are the centrosomes. So the microtubules arise from the centrosomes, right? So this is another that kind of microtubule that is arising. But there are microtubules that are not connecting to sister chromosomes at all. They are not connecting to sister chromosomes, sister chromatids. Therefore, we have two kinds of microtubules. We have two types of microtubules as kinetochore microtubules and non-kinetochore. Okay. So take a guess who are kinetochore. This is a kinetochore. This is a kinetochore microtubule. This and this, these are non-kinetochore. Right? These are non-kinetochore. Got it? Hammer microtubule doesn't get connected to sister chromatin. Okay. You don't need to draw this just for your understanding. Okay. So prometaphase. That is the next important thing that is happening in prometaphase. Some of the microtubules that attach to the kinetochore of chromosomes move the chromosome back and forth. Microtubules which are not attached to the kinetochore interact with those from opposite poles. Right? So you can put an arrow that says some of the microtubules that attach to the kinetochore of chromosomes. Put an arrow there and name those microtubules. What are they called? Kinetochore microtubules. Right? We have two kinds of microtubules as kinetochore and non-kinetochore. Online people, any questions? No. Kinetochore and non-kinetochore. We have two types. Then the microtubules which are not attached to the kinetochore, put an arrow there. Those are non-kinetochore microtubules. Non-kinetochore microtubules. And these two kinds of microtubules behave differently. That's also something you need to know. These kinetochore and non-kinetochore microtubules, they behave different in the way that they are either getting longer or shorter. Okay, but what kind is getting longer? Who is the dikvena kind? These non-kinetochore people are actually getting long. They are pushing each other. Right? They are sort of trying to move away from each other. Therefore, they are getting longer. Non-kinetochore. Right? Kinetochore are going to get shorter, like this. Kinetochore ones are going to get shorter. Okay. Why is that? Just focus on the non-kinetochore for a second. Why are they getting longer? Why are these non-kinetochore microtubules getting longer? Because they are the sort of the apparatus that is going to make the cell longer. Right? So in order to divide into two, the cell has to be long. Round cell like oval and divide them. Right? So that lengthening is actually done by this non-kinetochore microtubules. They are pushing the cells to be long. Okay? Clear? So that is all about prometaphase. So come to the tute. They interact means actually they connect with each other. Because why? There are two centrosomes. So these actual microtubules, they come as two halves and then they connect at the middle. Right? Because they come from the two opposite, let's say two poles, the centrosomes in the two poles, they meet at the center. That is what we mean by interact with each other. Okay? And that interaction can be different as I mentioned. So later that interaction between non-kinetochore is sort of pushing each other. Right? Let's come to the tute. Second diagram. for the topic. Second diagram for the topic. Yes, prometaphase. Prometaphase. Second diagram for the topic prometaphase. Then underneath we have mainly three things to write. Okay. First one, nuclear envelope fragments. Nuclear envelope fragments. You can see in the diagram, it's been it's been broken down into pieces, right? Nuclear envelope fragments, right? Second one, what is that? Number two, chromosomes get even more condensed. Chromosomes get even more condensed. Then underneath there, categorize microtubules into two. Categorize microtubules into two. Microtubules are of two types, as kinetochore and non-kinetochore. Arrows දෙකක් දාලා. Microtubules are of two types, as kinetochore, non-kinetochore. Okay. Moving on, any questions? Moving on, metaphase, right? Metaphase, very important phase. Okay, let's read. Come back to the resource. Okay, centrosomes reach the opposite poles. Centrosomes reach the opposite poles. This is again partly due to this non-kinetochore microtubules pushing them, right? Centrosomes are pushed away from each other because of this non-kinetochore microtubules, right? The chromosomes have arrived to a place called metaphase plate, which is located equal distance from each pole. The metaphase plate is at equal distance. If this is the cell, metaphase plate is at a midpoint of the cell, right? Metaphase plate. Because why? The cell is going to divide into two from that position, right? Cell division happens there. Metaphase plate. Okay, it's almost equidistant from each end of the cell. Okay. The centromeres of all chromosomes are located at the metaphase plate, right? Something nice happens. This is where, I mean, under the microscope you will be able to see metaphase very clearly. Because of this, all the sister chromatids are going to be arranged in a line at their sort of their all their centromeres are going to be at the same straight line. ඒගොල්ලෝ පොඩ්ඩක් එහෙට මෙහෙට කැරකිලා තියෙන්න පුළුවන්. Their orientation can be a bit not straight, but definitely their midpoint, their centromere is going to be at the metaphase plate. Okay, that is definite. So I forgot, put number one at centrosomes reach the opposite poles. Centrosomes reach the opposite poles, that is the first thing under metaphase. Centrosomes reach the opposite poles. The second thing is chromosomes have arrived at a place called metaphase plate. Underline the word metaphase plate in red color or whatever. Metaphase plate, very important phrase. Okay, metaphase plate. Okay. At the end of this phase, each chromosome of the cell get attached to kinetochore microtubule at their centromere and align at the metaphase plate, which is basically the same idea. Right? Clear about the metaphase? Come back to the tube, second page or next page. Both of those diagrams are metaphase. Both the two diagrams are metaphase. So put the topic metaphase as a common topic. Okay, දෙන්නටම common topic එකක් metaphase. In the first diagram, what is happening? Sort of the sister chromatids are still on the way to the metaphase bit. Can you see? The sister chromatids are still on the way. Yes. So in the second diagram, you can see nicely they have arrived. They are nicely arranged at what? If you can draw a dotted line with a whatever red color pen, no, draw a dotted line connecting all these centromeres, right? Connecting all these centromeres in the second diagram, draw a dotted line. Yes. What is that? Online people, what is that dotted line is indicating what? Metaphase plate. Yes. Okay. Metaphase. Autocorrect, right? Yes. Yeah. Then you can by yourself write down a few things happening in metaphase. මොනවද වෙන්නේ? ඔය තියෙන ඉඩ තියෙනවනේ. What is happening? Centrosomes reach the opposite poles. Right? You can write down a few things. Centrosomes have reached the opposite poles. Chromosomes have arrived at a metaphase plate. Then you can say centromeres are located in the metaphase plate. Few things like that. There's a bit of an issue with the printing, no? If you're okay with it, it's fine. Okay, metaphase. In the next diagram, if you look at your left bottom under metaphase, you can see the two kinds of microtubules as the kinetochore and non-kinetochore. Can you see in the below diagram? You can see the two kinds of microtubules, right? Yeah, in this diagram, this one, right? We haven't gone there, but can you see? There are microtubules that are connecting to kinetochore. Almost like in that diagram, most of the ones are kinetochore, no? But can you see there's two people that are not connecting to sister chromatids? Those are called what? Non-kinetochore. So draw two arrows. I hope you can see the two. There are two microtubules that are connecting with each other. We say interacting with each other. No? So label those two people in the below diagram. Okay, in the below one. There are in the metaphase diagram also there are few people that are non-kinetochore. No? Label them as kinetochore, non-kinetochore. What is important is non-kinetochore, right? Okay. Clear about metaphase. Only two more phases to go. Right? Before interval we can do anaphase. Okay, come to the resource. Anaphase. First event happening. Sister chromatids are separated at the centromere. Right? The cohesin protein is going to dissolve. Right? Cohesin ගේ role එක ඉවරයි now. What was his role? To keep the sister chromatids together until all this happens. Co-metaphase, metaphase, until all those things happen, the cohesin was just holding on to the two sister chromatids. I mean, holding the two sister chromatids together. Now its role is over. Now the time is to do what? To separate them. Right? දුවලා දෙන්නව අර let them go. Right? They are going to separate. They are going to be separated. So anaphase, first thing, sister chromatids are separated at the centromeres. Put number 1, highlight. Microtubules attached to the kinetochore get shortened and pull the sister chromatids towards the opposite poles. That's what I said. These kinetochore microtubules are getting shorter. They are pulling the two sister chromatids towards the two poles, away from each. Again, the මේ දෙන්නට so දෙන්න එකට ඉන්නකොට they are sisters. Sisters නම් they are chromatids. Then අපව ස්වාධීන වුණාම they have when they have become separated, they are called again what? Chromosomes. Okay. එකට ඉන්නකම් එක නම separate වුණාම එක නම. Again they are chromosomes. Clear? Cell elongates as the non-kinetochore microtubules lengthen. The non-kinetochore people are going to push the cell so that it becomes longer. Right? Number 2, microtubules attached to the kinetochore get shorten and pull the sister chromatids to the opposite poles. That is the second event happening. Third one, cell elongates as non-kinetochore microtubules lengthen. Cell elongates. Okay. By the end of anaphase, equal and complete set of chromosomes are found at each pole of the cell. Right? Listen, equally, we have divided the DNA. Right? We have nicely packaged them. We have combined them into a double package as the sister chromatids. Now we have separated the double package and given to the two sides. Right? Come back to the tute. This is the diagram of anaphase. Right? Right side, this one. See, from your aspect, left side, this. The diagram. Can you see? The sister chromatids getting separated. Right? So write down the things happening in anaphase. Number 1, sister chromatids are separated at the centromere. Sister chromatids are separated at the centromere. First one, sister chromatids are separated at the centromere. First one. Sister chromatids are separated at the centromere. Number 2, write down. Kinetochore microtubules shorten. Kinetochore microtubules shorten and pull the sister chromatids towards the opposite poles. And pull the sister chromatids towards the opposite poles. And pull the sister chromatids towards the opposite poles. Third one, non-kinetochore microtubules. Third one, non-kinetochore microtubules elongate the cell. Third one, non-kinetochore microtubules elongate the cell. Okay. Anaphase. Right. The two sisters are now separate and independent. Telophase. Come back to the resource. Final part of the M phase of mitosis. Final part of what? Nuclear division. Farmer cell like a divide. Only the nuclear division is coming to an end. So read telophase. First event, nuclear envelope reforms. Nuclear envelope reforms. Highlight and put number 1. Nuclear envelope reforms. Nucleoli reappears. Number 2, nucleoli reappears. Spindle microtubules get depolymerized. Number 3. Then we don't need the spindle anymore. Right? So, okay. What is the monomer of spindle microtubules? It's in your resource, right? Whatever microtubule is composed of what? Tubulin protein. Yes, 13 columns of tubulin form the wall of the microtubule, right? Okay. Go back and read about cytoskeleton. Spindle microtubules get depolymerized. Number 3. Chromosomes unwind and become less condensed to form chromatin. Number 4. ආපහු පස්සට යනවා නේ. Nicely pack කරපුව ටික පොත් ටික ආපහු එළියට ඇදලා දානවා. Right? Chromosomes unwind and become less condensed. Finally, two genetically identical daughter nuclei. Highlight කරන්න තියෙන්නේ කොටු කරන්න whatever the word nuclei. Right? Very, very important. At the end of telophase, what do you have? Two nuclei only. Five things that happened in the tute. The last diagram is telophase. You can see, I'll explain the diagram. Go home and write down the things that happened. Okay, resource එකෙන් බලන්න ලියන්නදී. Okay, what are the things that you can see happening? Nuclear envelope is reforming. No, when it was getting fragmented, it was the same, right? It is getting reformed now. And what else can you see? Your microtubules are no longer there anymore, right? Just the centrosomes are there. Because why? They have gotten depolymerized. And can you see slightly these chromosomes have gotten a bit longer, eh? That's because why? They are unwinding, decondensing, right? Unwinding and decondensing, so they are coming back to their normal state, that is chromatin, right? Telophase. Yes. Any questions? So once the two nuclear envelopes are formed, we have two complete nuclei, daughter nuclei. What is left to do? We have two nuclei in the same cell. We have to give the nuclei to individual two cells, right? That's what we are going to do. That is the sort of the next step to the cell division, that is cytokinesis, Right, come back to the resource, page number 41 still. The division of the cytoplasm starts at the end of telophase. Therefore, at the end of mitosis, two genetically identical daughter cells are produced. Right. There are two ways how this cell, whatever the cell, becomes two. Now we have the basic requirement. We have two nucleus. Controlling centers we have established. We have to just divide into two. In animal cells and plant cells, there are two different mechanisms. Right? I have given these two mechanisms in the tube. In animal cells, there is something called the cleavage furrow. Right? The cleavage furrow. Right? So that cleavage furrow is actually formed by, it's a, let's say, a motor function, right? There are different motor proteins involved. Name me a motor protein. Actin. So that is, actin is mainly involved, right? So actin, what actin is going to do is, this actin is sort of slightly, I mean, it gets attached to the plasma membrane at sort of the midpoint, like this, right? This actin is going to get attached to the plasma membrane at the midpoint, and it is going to pull it, pull the plasma membrane. So when you pull the plasma membrane, at one point the plasma membrane is going to meet at the center. It becomes two. Actin is going to pull the plasma membrane close to each other, so the plasma membrane connects and separates the two cells. That is what happens in cleavage form. Right? You can see. It's a contractile ring of microfilaments. We say, මෙහෙම වටේට around the cell at the metaphase plate, right? At the place where there was the metaphase plate, you can remember, right? Metaphase plate is what? Is equal distance from the two poles. So it makes sense that we are dividing the cell at the midpoint, right? So it's the cleavage furrow is the result of a contractile ring of microfilaments, mainly composed of actin. You know, microfilaments is composed of actin, right? So that's what happens in animal cells. In plant cells, it's a bit more simple because we have a outer structure that we can get the help to divide the plant cell into two. That is called the cell plate. Cell plate is sort of very closely associated with the cell wall, right? So there's a plant cell like this. The plant cell got bigger because of what? The two gap faces and everything. So you have the cell wall outside, right? And you have the two newly formed nuclei on the two sides. Right? So what's going to happen is there's going to be a new cell wall forming in the middle. There's a new cell wall forming in the middle, and later that cell wall is going to be completely connected. So you're dividing the cell into two by making a new cell wall. Right? This process of forming a new cell wall is called the cell plate. Right? Cell plate. So to make the cell plate, what do you need? It's part of cell wall. Cell wall is composed of what? Cellulose and hemicellulose and those things. Right? So who is making this raw materials to make this new cell wall? Golgi apparatus. Golgi apparatus. Golgi is like the hardware store. Vesicle. Those vesicles are going to come at the sort of the midpoint, make the new cell wall, separate the cell into two. Cell plate formation happening in plant cells. Okay, in the previous diagram in telophase, I don't see a nucleoli reappearing. Okay, yeah. Can you see a nucleoli there? No, no. Yeah, that is not included in the diagram. Okay, anyway, cytokinesis, right? Two ways cytokinesis happens, either by cleavage furrow or by cell plate formation Animal cells have cleavage furrow, right? Plant cells there is cell plate. Cell plate is formed by vesicles secreted by Golgi. Page number 42. Vesicle produced by Golgi, cell plate. Then quickly, why do we need to do mitosis? Why do we need to do mitosis? Firstly, it maintains genetic stability. Mitosis is very, very careful that all the daughter cells that it makes is exactly identical. I mean, it can be different in other ways. Mitochondria number, chloroplast number, how many Golgi, those things could be different, but definitely, DNA is exactly the same. DNA is exactly the same in the two daughter cells, right? That is one of the most important things that is assured by mitosis. DNA will run change and then, right? If mitosis took place properly, okay. Growth and development, right? So your bone cells underwent mitosis. Whenever you have a wound, your skin cells does mitosis. Your hair is doing mitosis as we speak, right? It's making keratinocytes. New keratinocytes are producing keratin. Hair is getting long, right? It's like that. Okay. Your liver is constantly getting, I mean, the liver cells are constantly dying and it's constantly making new cells. If you drink alcohol, your liver cells are constantly dying. But up to a certain level, your mitosis is trying to make new cells. Give me another example. In your stomach, right? Stomach is the best example. The stomach lining, right, is one of the most highly mitotic places. Every 3 days, roughly, there's a new stomach lining, completely new one established. Why? Because your stomach is exposed to HCl, no? About pH 2, 1 to 2 pH, burn venom. And also in the stomach, we have the enzyme pepsin. Pepsin, we talked about. What kind of a enzyme is it? Pepsin is a protein digesting enzyme. So, your cells are mainly composed of what? Proteins. So, our own enzymes are going to destroy our own cells in the stomach. There is no other option. If you want to digest the food, you want to secrete the enzyme, but the same enzyme is going to digest your own cells as well. That is called self digestion. So, what is the solution? You have to make new cells to replace the ones that are getting digested. So, mitosis is making new cells very fast in the stomach, right? Cell repair, replacement, and regeneration. That's what I told you. Whenever a cell is damaged, mitosis is going to make a new one, right? Replace curve. Okay. Particularly in wound healing, right? You have mitosis. Asexual reproduction. There are many animals that are reproducing asexually. Most of these cnidarians, sea anemones, some worms. Have you seen these planarians? They become two. Have you seen? That is called fragmentation and regeneration, right? In those animals, එයාට අම්මා තාත්තා කියලා මේ mother and father කියලා there's no requirement for two organisms. One organism is there, it can give rise to another organism asexually, very similar to mitosis. Asexual reproduction. Okay. So for your 2026, I'm today starting that is asexual reproduction. So for that, what you need is mitosis. Exact photocopy එකක් හදන්න. Okay. So those are the significances. Any questions? That brings us to the end of mitosis. After the interval, we can do questions. Yeah. No questions. Online people, any questions? Hmm? Take a break, 15 minutes. Come back at 10:50 when I come in now. 10:50. Okay. No, 10:45 is enough. 10:50. Line people, can you hear me? Okay, everyone is back now. So let's do a few questions from Mitosis just to warm up a bit. So if you turn back, there's going to you can find the questions. Um, did we do question number 5 that says there's 4, 4, and 5? Did we do that? No, right? Which of the following regarding eukaryotic cell cycle is correct? I'll project it on the screen as well. You can hear me, right? Out of the PQ arms, usually Q is the longer arm. Okay, chromosome. Okay? Usually Q is the longer arm. Right. Question number 5. Which of the following statements regarding eukaryotic cell cycle is correct? 2 minutes. So I didn't bother to change the question numbers because mostly I'm lazy. But more than that, you need to have an idea. So usually question number 4, 5, those are likely to be cell cycle or from the second lesson anywhere, could be from metabolism or whatever, right? Okay, online people, everyone is back, right? I'll project the question also. Question number 5 in the tute. Which of the following statement regarding eukaryotic cell cycle is correct? Okay, then what's the answer? Online people, send me the answer. What's the answer? Yes. Regarding eukaryotic cell cycle is correct. Anyway, we are studying only about the eukaryotic cell cycle because in prokaryotes you have this binary fission or something primitive, no? Crossing over, right. So it includes some meiosis, no? Without that, can't you get the answer? What is the answer? 5 then. Then okay, ignore the first one. I take a cut can, whatever. Crossing over says, anyway, it's wrong. Okay. Yeah. So discuss from the second one. Formation of chromatin occurs in G1. So they are, what they mean is formation of new chromatin. No, so there is chromatin. New chromatin occurs in G1. No, in S. So you can write a small review. DNA replication and formation of chromatin takes place in S phase, not G1. You can write your own review, right? Online people is clear enough. Shape. Hmm. DNA replication occurs in G2. No, it's again what something that happens in S. No, nuclear envelope reforms during cytokinesis. Cytokinesis is what? It's whatever the cytoplasm sort of divides into two to make the new cells, right? But nuclear envelope reformation is part of what? It's part of, so that happens in telophase, right? Not in cytokinesis. So you can write happens in telophase. This gives rise to two genetically identical daughter nuclei. Formation of mitotic spindle begins in prophase. Clear? Did you do this one? A response which correctly indicates the event and the phase in the eukaryotic. We did do it. No. Karadha? Yes. No. Question number 4. Which correctly indicates the event and the phase in the eukaryotic cell cycle. Okay, send me answers. Okay, good, good. I'm getting answers. Yes, what's the answer? Okay. 10 more seconds, and then we can discuss. Okay. Which correctly indicates. So usually, if they are asking incorrectly, they are going to bold it. Okay, correctly now they are not going to bold. Okay. Uh, Event and the phase. DNA replication G naught. No. Where? Right. Synthesis of proteins. Yes. I mean, that is the answer, right? Yeah. Yeah. G1 and also in G2, right? You can say also in G2 also there is protein synthesis. Chromatin formation? No. Yes. Production of organelles? I mean, could be either G1 or G2, no? Yes. Duplication of centrosomes? How? Yes. Where? Which phase? Huh? G2, no? Yeah. Specifically G2. Yes. Just the correct things as enough, right? No need to write a review, right? So correct everything except the correct one. Cut the wrong response and write down the correct phase. DNA replication in S, protein synthesis in both both gap phases. chromatin formation only in S, cellular organelles both gap, duplication of centrosomes, M phase. So you can see, right, they are asking very straightforward things in the paper, but the thing is, you have, we are in almost about halfway of the second lesson. So we have done about 5%, maybe, no, half of 5%, maybe 2% of the syllabus at this point, right? 2.2, 3% of the syllabus we have done. Right? Because why? We can only answer up to about fourth MCQ out of about 50. That's because of not what about the amount of sort of let's say weight of the syllabus, but because they are usually tend to ask more questions from this second lesson. That is to sort of equalize, because many students have done these first few lessons relatively well because they have the enthusiasm and මුල් කාලේ හොඳට පාඩම් කරනවා. So they are going to, out of sort of dukkha sympathy, they are going to ask a few more questions from these first, second, and third lessons, right? Otherwise, there's a distribution curve, right? Every year they are going to take samples from every district when they are doing the paper marking, and they are going to maintain this normal distribution curve, right? So every year it has to be sort of the number of, most of the students are going to be at average C B level, right? If this curve shifts to this way, that means most of the students are in FC levels. That means what? The paper is too difficult, right? Every year the same number of students should get the almost the same should get A passes, and also almost same number should go to F, right? So F number එක වැඩි වෙන්න බෑ. So if because of whatever reason in a year the FC numbers are very high, they are going to take marking scheme samples, sorry, marking answer scripts from different centers, and they are going to mark a few, and they are going to see මොකද වෙලා තියෙන්නේ ළමයි. If this curve has shifted this way, they are going to make the marking scheme less strict අඩු කළා, they are going to shift it back, right? And if many students have gotten sort of if the curve has shifted this way, they are going to make it strict. That means what? The paper was easy, no? The paper was easy. So they are going to make the marking scheme strict. මේ පැත්තේ hard, මේ පැත්තේ easy. So you don't know, right? So usually these kind of, I mean, what I'm saying is they give this some more questions from the early lessons to keep the sort of exam in the mid level, right? වැඩිපුර ළමයින්ට CS passes දෙන්න තමයි try කරන්නේ. CSB passes, right? They are going to reduce F and A's. Okay, right. What shall we do next? Any more questions that you can do that is from mitosis? You can do this one also, no? Try this 1 minute. Right. Shall we discuss? What's the answer? Two. Okay. Right. In the cell cycle, DNA synthesis takes place in G1. No. එකම ටික අහන්න පේනවද? Protein synthesis occurs in G2. Yes. Formation of spindle begins during metaphase. And that's something different. Spindle starts to form in prophase, no? Yes. So you can write formation of spindle occurs during prophase. What are the parts of the spindle? Aster and spindle microtubules. Centrosome, aster, spindle microtubules. These are all structured essay also. They can ask you what are the components of the mitotic spindle. Condensation of chromatin fibers takes place during S phase. Condensation, right? සමහරවිට ටක් ගාලා if you remember something about chromatin and S phase, මොකක් හරි chromatin වලට වෙනවා in the S phase, you miss this. Right? So condensation happens in where? Again, prophase. A little bit more of condensation in pro-metaphase as well. Further condense කියලා තිබ්බනේ. Right? But mainly prophase. Right? Not condensation. If they have said synthesis, then okay. Division of the cytoplasm occurs during anaphase? No. What happens during anaphase? Sister chromatids separate. You can write down during anaphase, sister chromatids separate. Okay. What happens in which phase does division of cytoplasm happen? Cytokinesis. You can put an arrow and write cytokinesis. Right? Shape. State two significances of mitosis. There's four. There's four. What are the significances of mitosis? Write down. And also do the next one. At which phase of mitosis would the nuclear envelope be fragmented? Online people send me answers. Significances of mitosis. Yeah. Wow. Yes. What else? Okay. Anything else? Repair, regeneration, asexual reproduction. Right. At which phase of mitosis would the nuclear envelope be fragmented? If you can remember my diagrams, prometa, prometa, no? Right? Right. Then we have an essay question that is from meiosis. So probably your homework. Yeah, all the others are from meiosis. Right. Let's get started with meiosis then. Any questions? Right. So meiosis is really interesting. Why do you say that? It is so interesting and also really complicated about meiosis. Yeah. What do you think? So meiosis happens in any sort of reproduction. Online people, you can hear me. Yeah. Any sort of reproduction that is considered as sexual, right? Male and female කියලා දෙන්නෙක් involve වෙන any reproduction we require a cell division before that, that is your meiosis, right? So usually animals and plants have sexual reproduction, right? You might think in plants, how is there any male and any sexual reproduction? Yes, there is, right? There are male plants and female plants. Sometimes the same plant has the male and female parts. You know this, right? Right? So the book says end of telophase division of cytoplasm occurs. No, I mean begins to divide in the resource. At the end of telophase, yeah, it starts at the end of telophase. So that what it says is cytokinesis starts at the end of telophase, right? Not that it's part of telophase, right? You get it, right? So cytokinesis is a separate process that starts immediately after telophase, right? So not to be confused, it's not overlapping. There's no use of having two nuclei in the same cell. So the cytokinesis is going to start immediately, right? Okay. So as I was saying, let's focus on this animal life cycle. What is going to happen? When I took the introduction to cell division, I said in a sexually reproducing organism, if you take their DNA, you have a copy. It's not a copy actually. Two counterparts, right? So you were produced by. I mean, you are sitting here with your own features, your height, your skin color, your hair color, eye color, everything is a combination of your father and mother. Yes, right? A combination. You can have your father's nose, mother's height, right? Different combinations. So altogether, how many chromosomes do we have? 46. 23 from what you got from your father, 23 you got from your mother. Right? Exactly half. So from blue, Blue I'm going to use for father. Father's chromosomes I'll use blue, and mother's I'll use red. Okay. So what I'm trying to say is these are sort of very similar to each other in their shape, in their size, and also in the position of their centromeres. Very similar. But the information they carry is actually different. Because why? They came from your father, this one from your father, this one from your mother. But they are identical in those aspects. They are equal in size, equal in the position of their centromeres, and also they carry almost sort of, let's say, the genes coding for the same thing. What is a gene? A gene is part of DNA, a DNA piece that is going to determine one feature. For example, if you can roll your tongue, that's a feature that is going to be determined by a gene. How many can roll your tongue? Can, can't, can. Right. It's either you can or can't. That is given by these intrinsic muscles on the side of your tongue. Right? Your tongue again. Either side muscle groups there. If you have the nerve supply to them, you can roll. Right? Or dimple cheeks, that is also this kind of it's a gene. So if you don't have that gene, these are additional things. If you don't have that gene, there's a piece of muscle missing here, right? So a piece of muscle missing means when you're smiling, that piece of muscle is going to pull on your skin and make a like a depression there. So it's actually a, let's say, a mistake, right? Error. If you have dimples, it's a error. Okay, right? Then I mean, so those are gene features. Your eye color, skin color, intelligence, height. All those are determined by genes. So these are what chromosomes. So these chromosomes have the genes of similar type, but they can be different from whatever the information they are carrying. So for example, in this place, let's say you have hair type, either straight or curly hair type. So for your father has curly hair, your mother has straight hair. Can you see? Different information, but they are deciding the same thing, hair type, right? So this kind of a pair of chromosomes is called a homologous chromosome pair. Homologous chromosome pair. In a homologous chromosome, one was from the father, one was from the mother, and they are equal in size, equal in the position of their centromere, equal in the type of genes they have, but could be different in the information they carry. Those are the features of a homologous pair. So if you take one of your cells, skin cell if you do this sort of chromosome arrangement, how many homologous pairs do you have? Altogether 46. How many homologous chromosomes pairs? Into 23 pairs වෙසිතුනයි. Yeah, one set of 23 you got from the father, one set of 23 from your mother. So and also these chromosomes are of different types, sizes. So there can be uda chuti atta diga in right. Now in very narrow but very long, right? These are all different kinds of homologous pairs. Right? One set you got from the mother, yeah. One set from the father. මේ වගේ එක sets you have 23 sets. This sort of knowledge you need to have. Alright. So how does your father make a cell? I mean, how does your father give you only 23? His cells have 46, no? If you take one of his cells has 46. How is he only giving you 23? That is a result of meiosis or meiotic cell division happening in his, let's say, the gonads or the testis, leading to the production of sperm. In your mother, ovary produces eggs. In the eggs, there is only 23 because the eggs were produced by the cell division of meiosis. Very unique cell division. Nowhere else in the body except in your gonads, right? Where gametes are produced. We call the sperm and the egg gametes, no? Right? Only in the places where those kind of cells are produced, we have meiosis. Nowhere else. Right? Nowhere else. So, if you take an animal, our life cycle is like this. Okay? I have included a diagram. So, we have a diploid organism. What do you mean by diploid? They have one set of father's chromosomes, one set of mother's chromosomes. Yeah, so they have two sets. Diploid, right? So this is a diploid organism. This diploid organism is going to do meiosis. Let's say this is a male diploid organism. So through meiosis, what is it going to produce? Let's say it produces N kela. There's a cell. Let's say this is a sperm. Male in the, right? So this, because of this meiosis, the set of chromosomes that this sperm has is going to be half. Okay, so we are going to do sexual reproduction. So there's another animal has to be involved producing what? An egg. Egg එක कोई sperm में कोई set වෙන්න එපා, right? So these two are going to combine. That combination is called what? Egg sperm set happening fertilization. Right. Do you know where in your mother's reproductive system fertilization happens? In the fallopian tubes, right? So if you draw your female reproductive system like this is the uterus, and there's a snake like this, and from there you have the vaginal canal, vagina, and it opens to the outside. So this is the uterus. It has muscular walls, right? And this is the top of the uterus, right? And from the uterus we have these two tubes that are mettara thick, two tubes, right, that are going, and usually sort of like here we have the ovary, right? So around let's say here is where human fertilization happens. Sperm එක එනවා මෙහේ ඉඳලා ලොකු ගමනක් එනවා. Egg එක එනවා මෙහේ ඉඳලා. They are going to meet here. Fertilization happens. So anyway, right? So that is what happens in usually humans. So one end from the egg, one end from the sperm is going to produce a diploid organism. Question. Called what? What is the name we give? A zygote. Right? This is a zygote. Zygote has a zygote is how many cells? Only one cell. So we all started life as just a single cell. Okay? Actually, the first single cell is our egg actually. Because why? The sperm actually goes I mean enters into the egg, right? So the first cell is actually our egg that has been given our father. So the father's sperm actually drills into the egg and becomes the zygote. So right. So now you have trillions of cells. Our diploid adult organism has trillions and trillions of cells. If you take a human, so what kind of cell division brought the zygote to our adult? What kind of cell division? Mitosis. Don't know guys story. So we have covered this cell division. I'm going to do this one. Yes. If you like, you can draw this quickly. Only this one. Uh, the sort of animal. I mean, let's say sexual reproduction cycle. You can say life cycle. Yes, oviduct is another name for fallopian tube. Very scientific. Okay, oviduct. Label it sexual reproduction cycle. Right. Okay. Any questions? Make a theory natube. I'm not going to trouble you with whatever the plants do. Plants වෙන පිස්සුවක් නටනවා, right? Plants වලට වෙනම මෙහෙම මේ haploid වෙනම එකෙක් ඉන්නවා. There's a වෙනම plant එකක් ඉන්නවා haploid. Right? Again, just not just a cell. There's a separate plant that is making this haploid sperms and everything. Right? There are two generations. ඒක ඔයගොල්ලන්ට කොහොමද A level syllabus එකේ තියෙන farm වෙන part එකක් plant life cycles. Right? So that's we are going to start maybe in 2 months time. Okay, clear? So this meiosis we are going to talk about. Okay, come to the resource book. Come to the resource. Page number 43. Page number 43. Alright. Sexually reproducing organisms undergo a different type of cell division called meiosis. Meiosis is a type of nuclear division which gives rise to four haploid genetically non-identical daughter nuclei from a diploid mother nucleus. Keep reading. Meiosis involves two consecutive nuclear divisions, meiosis 1 and meiosis 2. Meiosis 1 is a reduction division, meiosis 2 is similar to mitosis. Each stage consists of four subphases: prophase, metaphase, anaphase, and telophase. Right? So there is, we don't study about pro-metaphase, right? There are subdivisions on there. So only four subdivisions we are studying. Before meiosis, one cell is in interphase. During S phase of the interphase, DNA replication occurs. Right? So before meiosis also there is an interphase. Right? There is no such difference. Before whatever the sperm and the egg is produced, the cell has to grow. Right? G1, S, G2 or phases fully yano. Right? So there is an interphase before the meiosis that you have to know. Right? Meiosis. There are two main parts. Right? As meiosis 1 and meiosis 2. What is the summary? Right? I think I have included a diagram. So basically, if you say there's a diploid cell, it's going to undergo meiosis 1. Meiosis 1 is going to produce two haploid cells, half. From those two haploid cells, there's going to be two more haploid cells. So meiosis produces how many? How many daughter cells? That's another big difference between mitosis and meiosis. How many daughter cells are produced? Four. Four daughter cells are produced. So that's why we say meiosis 1 is a reduction division. That is a division where this diploid nature will be converted to haploid nature. Okay. Meiosis 2, I'll say it now, meiosis 2 is very similar to mitosis. ඉන්න කෙනාගෙම කොපිස් එකක් හදන එක විතරයි. Right? So meiosis 1 is a reduction, meiosis 2 is very similar to mitosis. Keep that idea in your mind from the beginning on. Right, from the beginning on. Clear? Okay. So this I have given in the tute. There's a diagram there summarizing meiosis. I forgot to tell you where to draw. No? Did you draw here? Where? In the back. Right. So this is a summarization of meiosis. Okay. You can see in that example. In the example I have given, what is the sort of number of chromosomes in the cell we are considering? For simplicity, we 46 consider as an example. So in this example in the tute, the cell we are considering, how many chromosomes does the cell have? 2n number is given. Yes, what is the 2n number? 6, right? Instead of our 46, we are considering a 6 chromosome cell. Okay. So I told you before whatever this meiosis, interphase is going to happen. Interphase, in the S phase of interphase, what is going to happen? DNA replication. So the 2n is actually going to become what? What? So we are going to double, no? So there is going to be about 4n, no? 4n on. Yes, so can you see in the first diagram? All the 6 have become double. Can you see? Altogether 12 yeah? Can you see 12? So then meiosis 1 is going to happen. At the end of meiosis, then count how many you have in that in the diagram that says daughter cells of meiosis 1? How many chromosomes? Count. I mean, separately. 1, 2, 3, 4. Yes. No. Consider a set as 1. එතන 3 තියෙන්නේ. Okay. Right? Sister chromatids දෙකක් තමයි attached. Right? So there's only 3 there. After meiosis 1, there's only 3. Can you see? And after meiosis 2, again still there is only 3. Right? Can you see? 6 became 3 after meiosis 1 and 3 is 3 after meiosis 2 as reduction only happens in meiosis 1. Yes. Alright. That rough idea you have to have in your mind as we go along. Let's move on to meiosis 1. Very serious phase of meiosis 1 is the prophase 1. Okay. So you know each of these meiosis, those two stages of meiosis has their own prophase, metaphase, anaphase, and telophase. Meiosis 2 also has its own prophase, meta, ana, telo. They are called prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, like that. Right? So altogether you have 8 subphases, right? That is why meiosis is very complicated in that sense. Okay. Let's read about prophase 1. Cell enters to the prophase from interphase. You know, before whatever the cell division, we have a prophase. No? Chromosomes begin to condense. Put number 1 there, highlight. Put number 1. First thing that is happening, chromosomes begin to condense. Dear students, your tutes will be delivered within next week. Yes. Once you have given your addresses to the website, your tutes will be, the process will start. Okay. Right. First thing to happen in prophase, chromosomes begin to condense. Mukut karanna bෑනේ if you have only chromatin, you get it right. To do anything, to do whatever, we need to have the compact form, that is chromosomes. Okay. Second thing, nucleolus disappears, begins to disappear. That means the nucleolus actually disappears, right? So put number 2, nucleolus begins to disappear. Right. Next step. Okay. Put number 3 and highlight. Next, the formation of a zipper-like structure called the synaptonemal complex by specific proteins hold the two homologs tightly together. This paragraph is a notorious paragraph, right? Okay. Let's start to understand. So what do you know about homologous chromosomes? Kohen 1. Yeah, no, chromatin begins to condense to chromosome should be the proper way of phrasing it, right? Yeah, that's what they are trying to say, right? Yeah, there's nothing more to condense in chromosomes. Yes, chromatin begins to condense into chromosomes is the better way to phrase it. Yes. So talking about homologs, homologous chromosomes, what do you know about them? In a homologous pair, one came from the father, one came from the mother. In humans, how many homologous pairs do we have? Pairs? 23. Alright? Okay. So in your tute, can you draw this? A few homologous pairs, and let's write down a few points about homologous. Yes, 23 homologous pairs. So just we can call them homologs. Under meiosis, draw a small diagram, few examples of homologs. Then let's write a few things. Remember, to be honest, they are not exactly like this, right? They are DNA, no? Right? So they are wavy like this, and the lower arm is also sort of wavy like this, and the other one is sort of wavy like this, and මෙහෙම ගිහිල්ලා තියෙන, right? So that's what the actual appearance is like. Okay, just to be neat, I'll show you a proper this sort of arrangement of homologous chromosomes. Any question? Okay, shall we write? Shall we write? Okay. Write down on the homologs a few points. That's going to in sort of you don't need to study these, right? But I'm saying that's just going to increase your understanding. That's why I'm just giving. Okay, you don't need to study this. What you need to memorize is the resource. But to memorize the resource, you need to understand what the resource is saying, right? Okay, write down a diploid organism. A diploid organism a diploid sexually reproducing organism. Okay, a diploid sexually reproducing organism. A diploid sexually reproducing organism has two sets of chromosomes. Has two sets of chromosomes. Has two sets of chromosomes. Next point. One set received from the mother. One set received from the mother, within brackets, maternal chromosomes. One set received from the mother, maternal chromosomes. Then one set received from the father, what are they called? Paternal. Paternal chromosomes. Next point. A homologous pair contains one maternal chromosome, one paternal chromosome, and one paternal chromosome. That's it. A homologous pair contains one maternal chromosome and one paternal chromosome. Next point. Those two chromosomes are equal. Those two chromosomes are equal in shape, size, position of the centromeres, and the location of genes. And the location of genes. Good. Next point. The information they carry, but the information they carry is different. Why is that? Don't write. Why is that? Because one came from the father and one came from the mother. If they are not very close relatives, very different information. But the thing is, because they are both humans and both male, one is male and one is female, they are going to have 46 and they are going to give 23 into the sperm or the egg, and in the fertilization process that 23 is going to pair up with each other, right? That is what we call as homologous pairs. What are the features? They are equal in shape, size, position of the centromere and the location of the genes. But the genes, the information in the genes can be different. What is the example I took? Hair type. The hair gene is in the same place in the two homologous chromosomes, but the information is different. Your father is saying my son is going to have curly hair. Mother is saying straight hair. So there's going to be an interaction between those two genes, and that's going to determine either if you have straight or curly. That depends on the gene itself. There are dominant genes and recessive genes, if you have heard. So the dominant gene is going to sort of press down the other one, and he's the one who's going to decide if you have curly. If your father's curly hair gene is a dominant one, then you're going to have curly hair, right? Like that. So that's about homologous chromosomes. I will show you how in genetic technology, gene technology, we arrange homologous pairs, like we sort of pair them up, right? It is called as a karyotype, right? This all the chromosomes in an organism nicely organized, it's called a chromosome, right? I have shown you this diagram before as well when we did the introduction to biology. I think in the very first class I showed you this, right? This is how it looks like. In a human, we have 23 pairs of homologous chromosomes. Can you see? If you take the 6, both the pairs are right. So don't be confused now, right? This is one homologous chromosome, this is one. But why is there two? Because they are about to go undergo meiosis, right? So they have two sister chromatids. ඒක හිතන්න එපා. This is one set. This is one chromosome. This is one chromosome. This is one chromosome, right? So you have 23 pairs, no? 21, 22, 23. Got it? මෙයා කවුද special XX sex chromosome, right? There's a special one. The 23rd one is special because that is going to determine if you are male or female. So I think you know this O levels වල තියෙනවානේ. The XX නම් that's XX chromosomes ആണ് තියෙන්නේ. Female. X, what is the male chromosome configuration? XY. XY. And I think I explained this. This is not a normal set of chromosomes. What is the problem with this? Yes, online people, do you see a problem with this? I don't think it's very clear for the online people. Yeah. Any abnormality you see? Count කරගෙන ගියොත් here there's a problem. තුනක් තියෙනවානේ. Only two, I mean a homologous chromosome set. We always say it's a pair, no? Pair නෙමෙයි මෙතන තුනක් තියෙනවා. So this is not normal. This is called Down syndrome chromosome. I mean karyotype. So they have 47 instead of 46. Yes, right. So it's going to cause Down syndrome. You have seen Down syndrome people, right? In the tute, I have even put a diagram. Right. They have characteristic facial features, right? Shooting a hayak and straight hair. Few distinct features like that, right? And most uniquely low IQ, right? IQ is ගොඩක් අඩු. ගොඩක් නෙවෙයි. I think there are normal people with their sort of IQ as well, right? But they are inherently disadvantaged, right? Okay. So I told you, like in your childhood, so you are born with the same number of neurons. So බබාට වඩා වට දැන් මොලේ තියෙනවා කියලා අපි හිතනවානේ. How do you have, if your baby version and you have the same number of neurons in your brain, what is different now? How are you, let's say, more smart now? Yeah, the neurons have gotten bigger, and because of the stimulation, particularly in your first 2-3 years of life, you were stimulated, ne? අර නිකන් අපි කොට්ටේ කුඩ එල්ලන්නේ කරකෙන් පාට පාව ශබ්ද දාන, right? Because of all those visual like movements and the sounds and everything that is going to your brain, the neurons are going to make new connections. Neurons are going to make new connections. They are going to make get bigger, and there is something called neuroplasticity, right? In the first few years, your personality will be determined and what kind of an IQ you have. There are people who are visually creative, who have like auditory creativity, intelligence. Some people can, like we say, some people can see sounds, right? There's a certain color they sort of visualize when they hear a sound, right? I think this Charlie Puth has that ability, right? He hears a sound and he gets like a visual picture of music, right? So there are different kinds of, let's say, neural associations depending on how gifted you are, and also what you call a stimulate picture with you, right? Okay. So anyway, what I told you was how these homologous chromosome sets are arranged. Okay, clear about homologs, right? Where were we? We were talking about prophase one. So we were here in the formation of a zipper-like structure called the synaptonemal complex. By specific proteins hold the two homologs tightly together, right? Most important part of meiosis is understanding this prophase one, and particularly this synapsis taking place, right? What is synapsis? So you know, let's take your mother's chromosome. So what is going to happen to your mother's chromosome before cell division? It's going to undergo interphase. After the interphase, is there only one chromosome? There's two. Because why? DNA replication. So your mother's chromosome has now a sister, that is called the sister chromatid. So this is now doubled, a pair of sister chromatids attached at the centromere by what? Cohesin protein. Yes, same thing is going to happen to your father's cells. Before they divide by meiosis, they are going to undergo DNA replication. So father's chromosome now has a sister chromatid attached at the centromere. Okay, if I say centrosome, remind taggala kiyanna, okay, that level. Centromeres are attached at the centromere. Okay, then in prophase 1, what's going to happen? These are going to do something very peculiar. Right? එතකොට මේ දෙන්න මෙතන හතරක්, right? These by, but we consider these two as the same. Why? Because the exact copy, right? So this entire unit is what? It's a homologous pair. What is special about it? They have doubled. Right? This is still a homologous pair. This is a homologous chromosome. This is a homologous chromosome. Altogether, it's a homologous pair. Imagine this is, let's say, මනුස්සයෙක්ගේ නම් 7th chromosome. 22ක් තියෙනවනේ. Right? So then, what does the resource say? There's a formation of a zipper-like structure. Zipper-like structure that is going to hold these chromosomes tightly together. It's like this. So I'll draw them like a boxy form. So it's easy to sort of show this, right? And here attached to this one, we have the other sister chromatid. Okay, right? So these are what? Mother's sister chromatids. These two are going to come together to form one unit, right? So the father's one is going to be sort of underneath, right? Yatting in or connected to this one, and the other one is going to be connected to this one, right? Let's yeah, how do you visualize this? Let's draw the other one underneath like this, right? Right. So the two fathers and mothers chromosomes are connected to each other by a zipper-like structure that is called, I mean, they are held together by a special protein that you don't need to require, I mean, you don't need to know. So that protein connection is in between, right? So maternal chromosome, that is now two sister chromatids, is connected to the paternal sister chromatids by special proteins. So this complex, I mean, to be exact, your resource says this connection of proteins, this green color connection is called the synaptonemal complex, right? So this connection is called as, I mean, you can also call it as synaptonemal complex, but what your resource says, this protein set of proteins, these green color proteins that are connecting the homologs together, there is a synaptonemal complex. Okay. So now what we have done in the prophase, sorry, before the prophase there was interphase. No, we have copied the DNA and we have two sister chromatids, mother's chromosome, father's chromosome. In prophase, what we are going to do is we are going to paste them together by the synaptonemal complex. Clear? Right. Come to the resource. Keep reading. Yeah. So the zipper-like structure called the synaptonemal complex by specific proteins hold the two homologs tightly together. So the two homologs are connected tightly by the synaptonemal complex. Then there's a definition. The pairing and physical connection of the homologous chromosomes is called synapsis. So you can say this, let's say this stage of the cell division is called synapsis, which is part of prophase 1. Which is part of prophase 1. Okay, I'll show you a diagram of this. My drawing skills are not that good. Right? During synapsis, part of the DNA molecule of the non-sister chromatids of pairs homologous chromosomes break, exchange and rejoin at corresponding point. It's better to show this using a proper diagram. Right? So what we have established here, clear up to right this now, right? Mother sister chromatids, father sister chromatids, these are homologous pair. They are connected by special proteins to form synaptonemal complex, which is holding the two homologs together. Okay. Yeah. Can you see? Right. This is what's going to happen. So you can do the centromere, right? And so this there's two paths, right? What are those? The paternal sister chromatids. There's two, right? This is maternal sister chromatids. So this is what? The maternal chromosome. This is the paternal chromosomes together, which has two sister chromatids, right? This is the centromere. Okay. Then next step is what? Synaptonemal complex forms. Green color, can you see? Connection between the two homologs, right? Paternal and maternal is connected to each other by the green color special proteins, right? Forming the synaptonemal complex. Then what is going to happen is non-sister chromatids break and exchange. Who are the non-sisters? Mother and fathers are the non-sisters, right? So mother are the sisters. So mother has two sister chromatids, that is sister. Then father's chromosome also has two sister chromatids, that is again two sisters. This exchange is going to happen between, if I say simply, red and blue, right? Red and blue. What is blue? Father. Red is mother. So they are non-sister. Always the step that we are talking about here, that is called crossing over, happens between non-sister chromatids. Okay, it's a very small thing. Can you see? So this is a place where this crossing over is happening. What they have, what's going to happen there is part of the DNA breaks and goes to the other side. Part of the red is going to break and go to the blue side. Part of the blue is going to break and come to the red side. Mother and father exchange information, right? Mother's homolog sends a bit of information into the father. Father's homolog is going to send some information into the mother. Cross, right? So at the end of the day, it's going to look like this, right? Okay. So there is a bit of blue in the mother's side and a bit of red. I mean, this is not very clear. There's a better one. After crossing over has taken place, it's going to look like this. Can you see? So this blue were originally completely purely fathers, but now this blue has a red piece. This completely red was originally purely mothers, but now it has a blue piece. That happens because of what? Crossing over. Crossing over. Clear? I'll explain again. Right? Originally, this cell had 6 chromosomes. DNA replication happened. S phase. It became 12. Then the 12 got arranged into what? Homologous pairs. Right? 6 homologous pairs. Then there was breaking, exchange and reformation between what? Non-sister. Non-sisters are what? Between father and mother. So can you see the places? So that places where that crossing over or this exchange happens are called chiasma. Okay, chiasma. So this is the situation after whatever this crossing over has happened in prophase 1. Any questions? I'll show this to you again using the software as well. So this is what we call as a gamete mother cell. It can become either a sperm or egg. Right? It can produce either a sperm or egg. Right? So there is, I think, condensation happening, right? And the condensation happen. Then let's remove this plasma membrane to be more clear. Right? Let's remove the nuclear envelope also. Can you see the blue color are your what? Paternal chromosomes, but they have been doubled. No, so they have 2 sister chromatids. Deක sister chromatids. You have the red ones also. Can you see? Doubled. Right? I'll zoom in a bit more. Right? Right? Can you see the red ones are doubled? Those are mothers. The blue ones are fathers. They are connected at their centromeres. Can you see? They are connected at their centromeres. What is the next step? They are going to pair up. Blues and reds are going to pair up. Can you see they are coming close together? And centrosome right? Right? The blues and reds have paired up. Online people? Right? Can you see? This is what is happening. Synaptonemal complex is forming. Blues and reds are pairing up. What happened? I selected meiosis 2. Hold on. Meiosis 1. Hold on. Alright, okay. I'm moving the layer now. Let's remove this nuclear envelope. So where were we? Synaptonemal complex. Can you see the blues and reds have paired up? Yeah. Then what should happen next? Crossing over, no? This exchange. Right? So exchange is going to happen. Mother is going to give some to the father's side. Father is going to give some to the mother's side. So it's going to immediately happen. So pay attention. Right? Let's see. They're still moving. Right. So it's a bit incorrect, but pay attention only to the chromosomes. And can you see there's a bit of something weird happening here, right? And they are getting very close to each other. And can you see? Apart from red, blue gear, blue red gear. Can you see? Crossing over took place. Yeah. Now it's a bit of a salad, no? Right? So the mother's chromosomes have a bit of father inside, right? There's a mix, right? If you look into the other parts, you can see this red one also has some blue, and here the blue, sorry, the blue one has some red, and if you take a look at the other chromosomes also, the blues have reds and the reds have blues, right? Yeah, everywhere you see crossing over has taken place. Okay, can you see? Right. Then that is the end of meiosis 1. Then separate, but we are still in prophase 1. Anyone who wants me to explain again? Prophase 1. Summary. What's going to happen? Before meiosis, as common to any cell division, interphase is going to happen. DNA is going to be doubled. Then chromatin is going to be condensed to chromosomes. Then the two copies of the chromosome are now called as what? Sister chromatids. They are attached at the centromere. Something weird is happening in meiosis. What? The fathers and the mothers homologous chromosomes are going to come close together. They are going to be very friendly with each other and they become attached by the synaptonemal complex. In the synaptonemal complex, there is something called synapsis happening. Synapsis is what? Includes crossing over. Part of the father is going to go into the mother's DNA, part of the mother's is going to come into the father's DNA. And that places where this crossing over took place will be later visible. Right? Those are called what? When there's one of the such places. Can you see? Here? Can you see? In the red one there is blue. That is this kind of a place is called what? This is also similar. Right? Here is also chiasma. Chiasma. Okay. නෑ දෙකම වෙන්නත් පුළුවන්. දෙකම වෙන්නත් පුළුවන්. For a නැත්තම් තව කෝල් මං ගැනීම තමයි දෙකම පෙන්නන්නේ නැහැ. Right? But blue cross වෙන්නේ නැහැ. වැඩක් නැහැ. අනිවාර්යයෙන් blue red. So what do we call that blue red exchange? Non sister, no? Non sister cross වෙන එක තමයි keep in mind. Okay? Never the same. Right? Sister cross වෙලා වැඩක් නැහැ. They are equally the same. Always non sisters cross. Non sisters means what? Father and mother cross. Okay. Right? අන්තිමට මේ අපේ ඇත්තටම at the end of meiosis, our target is to make this a scramble of DNA. Right? නැත්තම් ඉතින් අපිට පුළුවන් asexual reproduction කළා අපේ වගේම කොපියක් හදන්න. Right? That's the purpose, right? Two organisms get together to form a unique organism. Right? Unique organism. The next generation has to be unique. That's why we are doing sexual reproduction. So one of the ways of making the next generation unique is doing මේ වගේ පිස්සු වැඩ crossing over අර මේව කරනවා meiosis. All for what? Evolution. Evolve. Next generation එක හොඳ කර. Got it? Right? Because why? තව දෙයක් කියන්න. How do you evolve? Evolve වෙන්නේ කොහොමද? What is the basic idea of evolution? Strongest survives. Strongest means what? Not the physical strength. In the human world, what is our main strength has become? IQ. Right? Our intelligence is our strength. Right? So, in order to, what do you mean by, what is happening in evolution? At the end of evolution, what's going to happen? Stronger next generation, no? Next generation has to be stronger. That's why we are evolving. Right? So, in order to have a stronger generation, there has to be weak people also. Right? How do you know you are more smarter than someone? I mean, how do you know you are smart? Because there are dumb people. Right? There are people with low IQ. So, in the future, if we are going to increase in intelligence, what should we do? All the dumb people must be eliminated. Right? That is evolution. So, in order to know which one is dumb and which one is smart, That is called what? Variation. There are different intelligence levels in the society. So, evolution will say that only the smart will survive. So, to evolve, you need what? Variation. ඔක්කොම එක වගේ මොලේ තිබ්බනම් කොහොමද next generation එකට මොලේ යන්නේ? එහෙම බෑ. Variation තියෙනවා. Variation එකෙන් කියන්නේ smart people will get the chance to reproduce, right? Smart people will get the chance to reproduce. So their smart genes will be transmitted to the next generation and the weak people will die without making babies. Okay? That is evolution. So in a, let's say, in a normal, like a, let's say a forest ecosystem, only the strongest predator will live. මොකද යාට විතරයි කැම හොයාගන්න පුළුවන්. Right? So the strongest predators will survive and they will make children and those children will make even more children and the strong people will survive and the weak small predators are going to die without reproducing, right? So the weakness will be eliminated. Weakness is eliminated. So what happens in COVID? All the people usually with low immunity and also low financial status, right? They couldn't afford ventilators or hospital facilities. They died. About how many people? 2-3 million died, right? That is part of evolution, right? So in the human civilization, our strengths are what? Usually intelligence. Physical strength is actually now, the value is now less, right? Physical strength you can survive, right? Usually intelligence and maybe money. Those are your strengths. So to make money, you need brains and so that is evolution happen, right? Every day. That is because why? There is variation. There are dumb people, strong people, weak people in the society. So this variation comes from different things that are happening in this meiosis, actually, right? Sexual reproduction එකේ වෙන මේ වගේ different changes in the you have variation. Okay, right. Profess 1 clear. I think we have only time to cover meiosis 2 today. Sorry, meiosis 1. Any questions? No. We have 10 more minutes, right? On the other hand, yeah, let's finish Profess 1 only, okay? Right. So where were we? This process is called crossing over. The points of crossing over become visible as chiasmata after the synaptonemal complex disassembles and the homologous chromosomes move slightly apart from each other. There has to be a move there in the resource. The homologous chromosomes move slightly apart from each other. Nuclear envelope breaks. Centrosomes move toward the opposite poles, forming spindle and in animal cells. So that is something common to mitosis. Spindle formation normal විදියට වෙනවා. Kinetocore of each homolog attached to microtubule from one pole or the other. The homologous pair then moves towards the metaphase split. So those are similarities to mitosis. Okay. Right. Then, so we were supposed to do this paper. I was planning to finish meiosis 1 almost, but as 5 minutes. Can you do the mitosis part of it? Yeah. Online people in the group, I put a paper 6. Can you open that? Online people, paper 6. Can you open the PDF? Quickly try the first side. The second side is from meiosis. You can do this in 4 minutes again, right? Online people, are you able to access paper 6? Hold on, I'll send again. I'll put it in the chat. Hold on.
Recording in progress.
Okay, I sent it. Okay, I sent it to the chat, Zoom chat. Are you able to open? Please check. Paper 6. Paper 6, first side. Yes. Online people, you are able to access, right? Yeah. So you are supposed to label all these numbers. Huh? 1, 2, 3. Very easy, right? Yeah. Other side is meiosis. Okay. Okay. Answer who's still right? Yes. Okay. Let's discuss which type of cells divide via mitosis. I gave this last week. Yeah, somatic cells. Somatic, right? There are two types of dividing cells, right? Or you can say in your body there are body cells, dividing body cells. There are somatic and there are gamete mothers. Again, the gametes හදන cells. So mitosis වෙන්නේ your somatic cells, your skin cells, liver cells, uh, gastrointestinal system, wherever normal mitosis happen වෙන ඕනම තැනක ඉන්නේ somatic cells. Gamete mother cells are only in your male reproductive sort of gonads, testes, and ovaries. Okay, great. What are the names of the 5 stages of mitosis? Just to remind, right? Leander. New cells are produced through cell division. However, before cell division can occur, the cell needs to grow and duplicate its DNA. The phase before mitosis begins, where the cell spends 90% of its time, is called interphase. Simple. First diagram, what is number 1? That is, you can say plasma membrane or cytoplasm or whatever, both is okay. What is 2? Chromatine, right? Condensation is almost happening, but started to happen. It's still chromatine, no? 3, centrosomes, duplicated centrosomes, you can also say centrosomes. Second diagram on the right side, number 1, nuclear envelope. Number 2, sister chromatids is good. Sister chromatids. Number 3, nucleolus. So the word list has been given also. Don't use that. What is number 1? That is in the lower diagram. We have number 1. What is number 1? Non-kinetoco-microtubule, right? Number 1 in the lower left side diagram, non-kinetoco-microtubule, right? Is an issue. Number 1 is non-kinetoco-microtubule, right? Why? Check. Yeah. Yeah, no. Microtubule or kinetoco-microtubule, anything, right? If you say, I think it's going a bit udding again, but yeah. If you say it's kinetoco, it's fine. It's the way you can see it, right? Then 2 is what? Definitely. 2 can occur. Where the kinetoco-microtubule is attached. That is what? That is kinetoco. Can you see a small bump effect? That is the kinetoco protein, right? That is a kinetoco protein. 3 is actually Q arm and the P arm. 4 is P arm and 3 is Q arm. Not necessarily, right? Next diagram. What is number 1? Daughter cells. Daughter cells. In the above one, what is happening as number 2? Cleavage furrow. Cleavage furrow of cytokinesis. Cleavage furrow of cytokinesis. Any questions? ඔබ තවලත් තියෙනවා Right. Then we can finish for the day. Online people, any questions? Oh, sorry, I didn't see what is synapses. I'll explain next week also. Okay, what is synapses? We anyway start with a sort of a reminder. Okay, you can go. You finish it. Little bit. Oh, muka sport type. Right. Ah, pata same. Kalla 2027 खेला से होगा. Okay, Devi ne. Right. Okay, my kan. Protein mark is king and paper 5 ne. Apathy paper 5 ke liye. sama ටික ගේන්න next week මම ගාව තිබ්බා. මට enzyme එක හම්බුනේ. enzyme social එක හම්බුනේ නැහැ. තව practical seminar එක කරානේ. ඒ ටික මයිලස් එක කරේ නෑ enzyme එකට අයිති ඒක. ඒතර enzyme එක ඕනේ වුණාට proteins එක හම්බුණා. proteins එකයි මේකයි විතරයි ඊට පස්සේ. ඊට පස්සේ main tube එක. main ඔව්. නෑ නෑ මං කියලා දෙන්න next week. right. Okay. Okay. Thank you. You're welcome. Okay. Thank you for joining.